This presentation on QTLpoly is sponsored by the Tools for Genomics-Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 15, 2021.
So hello, everyone. It's kind of impressive that we still have so many people here in the fourth day. So thank you for attending this section. My name is Guilherme da Silva Pereira, but you can call me Gui for short. I'm currently working as a molecular breeder at the International Potato Center, as Dave just mentioned. I was supposed to be in Kenya, but because of the pandemic, I'm actually talking to you from Brazil. We are going to talk about another package called QTLpoly. This R package can be used for multiple QTL mapping in full SIP families of Polyploid species. We started this work during my postdoc at North Carolina State University under Professor Xiaoban Zheng's supervision. And it was part of the Genomic Tools for Sweet Potato project funded by Bill and Melinda Gates Foundation. So before we jump straight to the package, we'll see some of the underlying theory that is the basis of our QTL detection approach, which is the variance component model. This is important because we treat the QTL as random effects. So we'll talk about that too. I'll show some applications that will help us to understand some concepts better. And after some final considerations, we'll go through a short demo, all right? So this is supposed to be a 30-minute theory and 30-minute demo. So in the previous days of this workshop, we were talking about molecular markers and how we use these markers to build linkage maps, right? So we turn our attention to the phenotypes today. So when we collect phenotypic data in field, greenhouse, or lab, one of the first things we do is like to plot a histogram, right? Where we can see the distribution of these data in classes. And if there is some variation or not. One way of describing phenotypic data is through its mean and variance. In this example, we evaluated foliage maturity for a population of 156 individuals of tetraploid potato. And we can see that it means around 70, with the phenotypes ranging from below 40 to over 120. So what we are going to do here is variance component partitioning that I'm sure many of you are familiar with. Okay, we understand from the histogram that there is some variation, but what makes potatoes early maturity or late maturity? First, let's suppose we planted an experiment in the field with clones of Atlantic only, one of the parents, right? All the phenotypic variation we see is due to the environment, right? Because we have clones of a single cultivar only. Now, let's say we crossed Atlantic with another clone, in this case, B18295. So in addition to the environmental variation, the phenotypic variation we see in the progeny also has the contribution from the genetic component, right? Which is the contribution from the effects of several genes segregating in the population. Now, being able to estimate the variance component, the genetic variance component, means that we can compute a measure called broad sense heritability, which is very important for breeders. And we can go a little further by estimating the additive genetic variance with help of a measure describing how related a pair of individual is, either based on the pedigree or based on the genome-wide markers. And that's the kinship discussion that was going on. When it's often called, when we base these estimates on the genome-wide markers, it's often called the realized genomic relationship matrix, right? Either way, we are looking at the covariance between individuals to share heritable effects. With the additive variance component in hand, we can now estimate the narrow sense heritability, right? So we have here an example in which we take all the segregating markers in these potato full seed population and compute the overall additive relationship between seed pairs. And what we find is that the marker-based estimate is around the pedigree-based expectation of a half for full seeds, right? So there is nothing actually new here. It's just like to show you that this is actually matches with the expectation, right? The marker data. Now, what if instead of looking at all markers at the same time, we look at more specific loci? By doing so, we could basically find some regions that contribute to the variation of the trait of interest, which turns out to be the very definition of a QTL, right? In order to find the QTL, we look at the relationship between seed pairs in specific locations along the genetic map. So these locations are often called intervals. Our expectation is that in the region of a QTL, correlated effects are because of shared alleles identical by descent, and that those more related individuals look more alike phenotypically, right? Let's see how we identify more or less related genotypes based on shared alleles identical by descent in a full seed family. Starting with a diploid species, we have two different gametes per parent, and this is an outcrossing species, right? Highly heterozygous, so we can expect that these are four different alleles. And the gametes leads to four different genotypes when these parents are crossed. So these genotypes can share 0, 1, or 2 alleles identical by descent, right? So in tetraploid species, we have 6 different gametes per parent and a total of 36 different genotypes. So I'm not considering double reduction here. And a genotype pair can share 0, 1, 2, 3, or 4 alleles identical by descent, right? In the hexaploid species, which was the species we started working with, there are 20 different gametes per parent and 400 possible genotypes, right? So a genotype pair can share from 0 up to 6 alleles identical by descent. It's easy to see how higher ploid levels complicate things, right? We can see that some genotypes are completely unrelated for a specific locus, and some are completely related, or that the covariance among seed pairs is different depending on the alleles they inherited from their parents, right? In that specific location. From experimental data, we are not so sure about the genotypes of each individual in the population, right? Because we only know the dosage, or because of missing data, or because of errors. So that's why we rely on the probabilities, on the genotype probabilities we estimate from the map to find out how related a pair of individuals is. Now, one way of looking for not only a single, but multiple QTL, so Peter just showed us one way of doing it, and that's using a fixed effect model, right? So we are modeling the effects of a QTL as fixed effects. But one way of doing it is by searching for depositions where there is a significant contribution to the total phenotypic variance. So I'm looking at the variance component specifically here, right? And the identification of such regions is facilitated by the relationship based on alleles identical by descent in a certain interval, or as we just mentioned, right? So we are looking at a specific position. And of course, by adding these different variance components associated with different positions, we can have a multiple QTL model. The estimation of these variance components associated with QTL allows us to calculate the proportion of the total variance explained by each QTL, which is the very definition of QTL heritability, right? This model has been called random effect multiple interval mapping, or REMIM. And this is just another way to write this multiple QTL model. But some people would find it here easier to understand it in terms of variance components. So that's why I showed that first. And we can do so because the QTL are treated as random effects here, right? Note that for such a model, we only need to estimate one parameter per QTL. And that's why it's kind of easy and not so dangerous to do to put together multiple QTLs in the same model, right? Because for the random effect model, we only need to estimate one parameter per QTL, which is the variance component. But for a fixed effect model, we need to estimate, let's say for a tetraploid, we need to estimate for each QTL 6 effects, right? And every time we add a QTL, we need to estimate more 6 and so on. For a hexaploid, we need to estimate 10 and 10 every QTL. And for a octoploid, we need to estimate 14 and 14 for every QTL, you know? So that's why we decided for this kind of approach, so we don't get into trouble when trying to estimate several parameters at the same time for each QTL. So this model has been fully described in this publication in Genetics, along with details on the genome scanning and the hypothesis testing and a model selection algorithm. In general, we look for a QTL every centimorgan along the genetic map, and we add a QTL to the model one at a time, right? So we can test the next QTL conditional to these other QTLs already in the model. And this has to do with the increase of power that Chris was mentioning in the first presentation, right? And we declare a locus of a QTL when the variance component associated with it is significantly higher than 0. So we also change the hypothesis testing here, because for fixed effects, we are interested in means, and in the random effects, we are interested in variance components. We use restricted maximal likelihood to estimate the variance components, and score statistics to test the significance of the variance components based on a certain critical value, right? This critical value can be defined from a resampling method, also detailed in the paper. So it's kind of a permutation, but it's not exactly the permutation, but it's the same idea, to have this value of the genome-wide significance level. So a model selection algorithm we've tested and used has 3 steps, and Peter just showed us a way of doing it more manually. And actually in QTLpoly, you can do it manually, but we have an automatic function to do it for us, and it follows us this. So first, a model starts with no QTL. Second, a forward search adds QTL at a time, one QTL at a time, using a more relaxed genome-wide significance level of, let's say, 20%. Third, we reevaluate every QTL in the model and see if it needs to have, its position updated, or if it has to be dropped under a less relaxed significance level of 5%. So we allow more QTL at first, but then we test them again under a more relaxed significance level, all right? And we iterate through steps 2 and 3, both under the significance level of 5% now. So 20% we just use for the first round of forward search. And then we go more stringent with 5% until no more changes are observed in the model. And this is what we call a stepwise procedure, right? Okay, so now let's see some applications, starting with a tetraploid potato population obtained from a cross between Atlantic and B1829-5. The full seed population has 156 individuals, and that was something that Marcelo showed yesterday. And also in the first day, we are calling these SNPs from this population. We are building the map for this population. Now we are looking for QTL, right? And we phenotype this population for 7 traits across 4 years, and all details can be found here on this preprint. So again, the population is genotyped with a chip array with 8,300 SNPs, and the linkage analysis resulted in 12 linkage groups with over 4,200 SNPs. Using MapPoly, we identified all parental homologous haplotypes, which allowed us to, track the homologous parental contribution for each individual. Of course, due to recombination and segregation, each individual might be more or less related when we look at a specific location along the genetic map, right? So again, A, B, C, D are alleles from the first parent, and E, F, G, H are alleles from the second parent. For a tetraploid species, each parent contributes with 2 alleles, right? So, for example, this is how the genotype probabilities are distributed across the population. So in the x-axis for a specific marker, and we have the genotypes here in the y-axis, right? Some genotypes are represented, in this population 3, 4, 5 times, like the one at the top, A, B, E, F. But some of our other genotypes are not represented at all, such as C, D, G, H, right? Another way of looking at these probabilities is across the linkage group for a single individual, right? Here we can see the exact points where the recombination events happen. And the differences between individuals, again, will come through recombination and segregation, right? So every individual will have a different profile of genotype probability. And here we have the additive relationship matrix computed based on the genotype probabilities at a specific marker on linkage group 5. And I'm going to talk about this location soon. We also plotted the phenotypic values here at the top and on the side. Now, this is pretty disorganized, right? So if we order the phenotypic values and reorganize the relationship matrix as such, we can note that more related individuals are kind of grouped here by their phenotypic values, right? Either if it's high phenotypic values or low phenotypic values. Similarly, individuals with contrasting phenotypic values like low yellow phenotypic values and red phenotypic values show lower related events, right? This kind of behavior actually shows that there is a QTL here, or an association between genotype and phenotype, right? So in fact, this is the QTL peak location of a major QTL for foliage maturity on linkage group 5. And all details on these analyses can be found in the preprint I mentioned earlier. Going now to our next population of outer polyploid sweet potato, right? So it's another full seed family from the cross Beauregard by Tanzania. Beauregard is orange fleshed and Tanzania is cream fleshed cultivar. We also phenotype this population for several traits, and here we are showing some quality traits, right? Dry matter is positively correlated with starch, and beta carotene is positively correlated with flesh color, and starch has a negative correlation with beta carotene, right? In this work down here, we wanted to understand the genetic basis of these correlations. So this is more to show the stepwise procedure we use to detect QTL. So the same way we do in the fixed effect interval mapping that Peter showed us, we test every centimorgan position to see if there is a QTL or not, right? And here we have the intermediate plots we get from QTLpoly when we are running the REMI model, or the random effect multiple interval mapping model. So first we test every centimorgan, as I mentioned. Then we start to add significant QTL. So here is the first, and you can see that the profile changes as soon as I add the first QTL, right? And this keeps happening, so the profile is changing, and I'm including QTL under a lower or a more relaxed criteria, a genome-wide threshold, right? And then in that third step, we are like reevaluating each QTL that we included in the model to see if it holds under the multiple QTL model, right? So the testing of these QTL is actually conditional to all the other QTL in the model. So you can see that we can we keep adding, we keep confirming that these QTLs are true. Some of them are dropped, right, because they go below a more stringent threshold. And then I go, and then I end up with 3 QTLs here, right? And then I refine my profile to look for another QTL. And now I'm keeping the threshold high, right? Okay, so I can see that the 3 are still here after the refinement. And when I complete the genome, I can see another QTL here. So I added, actually I add this one first, and then I cannot add anything else. And then I again test every single QTL conditional to all the other QTLs in the model. And I realize that all of them are significant under this higher threshold, right? So this is the final QTL profile, and the QTL peaks are represented here by triangles, right? So once we have the final set of QTL, we can fit the multiple QTL model again, which will give us the estimation of each variance component and the heritability for each QTL. So in this case, 5 QTL explain 68% of the total phenotypic variance for dry matter in this population. And I can sum these heritabilities because they are fitted together in the same model, right? That's something you cannot do when you're doing single QTL analysis. We also found QTL for starch, beta carotene, and flesh color to be co-localized with the major QTL for dry matter on linkage group 3 and 12 consistently. So it's pretty clear that they share this region. And it has actually a nice explanation for it, but based on RNA-seq and things like that, but I'm going to focus on the additive allele effects here that actually show us that this is happening, or this negative correlation is happening because the exact alleles that are contributing, for example, to increase beta-carotene or the flash color score is the same that is decreasing the dry matter or the starch content, right? So we basically go down to the explanation of how this is happening just by looking at the effects that we estimate from this model. And again, the whole story can be found in that paper I showed you before. Now, just to end this section, for now, we've decided to run some simulations to compare fixed effect and random effect model. And I have to say that this fixed effect is just the interval mapping model, right? It's not the composite interval mapping. And we call it FEIM, right? Fixed effect interval mapping, in opposite to random effect in multiple interval mapping. So we use the bodyguard by Tanzania population genetic map. So we simulated 1,000 phenotypes, each phenotype with 3 QTL with different heritabilities each, right? So 0.3, 0.2, and 0.1. The QTL were distributed along the linkage map under 3 different scenarios. We either forced at least 2 QTLs to be linked, or all of them to be unlinked in the 2 extreme scenarios. And the third scenario let the QTL be allocated randomly. So the QTL could be linked or not. Then we ran both fixed effect interval mapping and random effect interval mapping with 5 genome-wide significance levels obtained through permutation in the case of fixed effect model, and resampling of score statistics in the case of with phenotypes with no error included. We expect 100% of power, which is exactly the case for REMIM. We also observed that the random effect model has relatively higher power and a better false discovery rate control than the fixed effect model for this case. This is especially true when it comes to the linked QTL scenario, right? Where the fixed effect model stays around 60% of power, whereas random effect model can be up to 70 to 80. So being more conservative leads to a relatively lower power, but it also decreases the false discovery rate, right? So with the REMIM model, we've been using a genome-wide significance level of 5%, which provides a relatively good power while keeping FDR lower than 10%, right? Of course, you can imagine that the downside of a multiple QTL model is the computational time, because you have to go through all those steps. We are doing model selection after all, right? Which comes with iterations and things like that. However, if you have to think about the benefits that this type of analysis might bring, right? Which is the context for increasing power, but also for separating linked QTL and for testing QTL by QTL interactions, for example, because we have all the QTLs in the same model, right? So QTLpoly can be found on GitHub, where you'll also find a tutorial and the right space to report any issues. With 4 main functions, one can have, their full analysis done. And on top of that, we added, print and plot functions as well. So that's what we are going to see just in a few seconds, all right? Some things I haven't mentioned yet include the ability to run fixed effect model and permutations inside QTLpoly using the same output from MapPoly. You can also include weights to the phenotypes, which turns out to be very useful when your adjusted means are coming from very unbalanced experiments. Finally, we've shown a model selection algorithm here, but QTLpoly is flexible enough to allow you to add or drop QTL the way you want. Of course, one should be careful when you use these functions. It's part of our to-do list, the idea of allowing genotypes from double reduction events, fitting multivariate models in multiparental populations, and also adding experimental effects for one-step analysis. So far, we actually expect that the phenotype data that we use in QTLpoly is analyzed prior to entering the QTLpoly pipeline, right? So you basically obtain your adjusted means or blues before you enter this pipeline. And of course, the list is endless, and we have the idea of implementing these and other things, but we only do it by demand, So if we need a list that it's not implemented yet, we just implement and make it available. So if you have any requests, you can always reach out to us. That we'll be glad to see what we can do. As always, we couldn't have done this work without great collaboration. So here is my thank you to Xiao Beng and Marcelo, and to Craig Angel, Bodhi Olukolu, and Dorcas Gemini, who provide the first data sets we actually showed here today. We were all actually just a small part of the GT4SP project funded by Bill and Melinda Gates Foundation. And thank you all for listening. So what we are going to do is, go to the polyploids.org webpage under tools and workshop QTL analysis. And the second link is the tutorial we are going to try to run today. And hopefully you'll have QTLpoly installed. If you install it before, like if the installation is, let's say, a week old, you might need to install QTLpoly again. Just running the last command line. So I'm going to try to do this with you. So what I'm going to do is copy the code here, paste into a new file. So let me make it bigger. File, new file, script, paste here. So that's what we are going to do. I hope some of you are with me on this. So I'm going to load QTLpoly, right? The data for this demo comes with the package. So this is genotype probabilities from MapPoly from yesterday's potato population. But here I selected only 3 linkage groups, so we can make it by the end of 20 minutes or so. And then I load the phenotypic data, and I can see what my phenotypic data is here. So we are dealing with 3 phenotypes, right? Foliage maturity evaluated on in 2007, 2008, and 2014. And here we have in the rows, we have the individual stages, right? We can actually do some kind of exploration here, like box plots, as Peter showed us, just to have an idea. And we can see very simple for the linkage group 1, that's the probabilities, and for the first individual, so I'll have something like this, and I can see it for the second individual, and so on. All right. This is just like in Poly QTLR. This is an array with 3 dimensions, right? So one for the genotypes, other for the positions in the genetic map, and the other for the individuals, right? Okay. So I'm going to run this function, and what it does is it gets data ready for QTLpoly. So I have here the genoprob object and the pheno object. You need to make sure that the names of the individuals match in both objects, right? And here we also select the diploid level, which is 4 for potato, and the step size, which is the how often we are going to test if there is a QT or not. So in this case, we are doing the test every, or we are going to do the test every 1 centimorgan, right? So once you get your data ready, it outputs some information here, but if you want more detailed information, you can use a print function. So it shows how many positions we are going to test in total, and for each linkage group. Also, how many individuals, non-missing individuals I have for each trait, right? Now, to be able to detect the QTL, we could use this same object, this data object, to run this score-based resampling method, but we are not going to do it here because we don't have all 12 linkage groups, and also because running the score statistics 1,000 times for each position would definitely take some time, and we don't have the time here. So I ran it beforehand, and I'm going to use the, the pointwise significance level here derived from this analysis. I can actually show you in the tutorial how we run this method, and of course, this needs to be done for every population you have. So this is not fixed, a fixed value, right? So 20% genome-wide significance level is 10 to minus 3, and 5% genome-wide significance level is 10 to minus 4, right? So then we have this automatic function for doing the forward and backward procedures for us. Again, as I mentioned, you can do this manually, but this is just implemented in an automatic way here, just to be a little faster. But of course, you can explore more later on your own. So again, we are going to need the data, right? We can select the column of the phenotype I had earlier. So here I have only the phenotype 1, but we can of course do the 3 phenotypes altogether. I mean, 1 at a time, but in the same command. Here we have the window size, which is the region you exclude around the QTL, or around the QTL already in the model. So this is where it comes the significance level we just described, and this is the d-value for estimating the support interval. So 1.5 gives us the 95% support interval. But if you choose 1, for example, there would be 99% and something like that. 2 would give you 90%. So it's actually up to you how far you want to go. And this is the number of cores you might want to use to run your analysis. I recommend you, if there is anyone following, you run only 1 phenotype and in one cluster, in 1 core, I'm going to go ahead and change these for 16 and these phenotypes to go through all 3 phenotypes we have. And I don't recommend you do this because it's likely that we don't have 16 cores in your computer. Right, so I'm going to start running these, and I can actually show, I'm using Linux here, right? So if I type htop here, you're going to see 16 cores out of 24 being used for this calculation. So that's how we can do it faster, especially using, a high computing system that you might have. So we are almost there. So I can go actually through the first phenotype. So far, at first we had, and each line is a step, right, of the process. So we selected first a QTL on linkage group 2 at 26 centimorgans, and here we have the position number of the on the map, and then conditional to this QTL on the in the model, we detected another QTL, which is this one linkage group 3, and then the package says no more QTLs were found, but there is a putative QTL here, so this is where the manual-based functions can be used. Maybe you decide to include that position if you have a good explanation for it. But we are not doing it here. And then it refines the position to see if both QTLs are kept in the model. And in this case, they are, and even the position doesn't change. And for the second trait, the same thing. Now we have a third QTL on linkage group 1. And then the refinement might change some of the positions, which is fine, because remember, this is always conditional to one QTL in relation to the other 2. And then the third QTL, the same thing, right? 2 QTLs only, and the positions that have changed. So there is a nice print function for that. So you can actually see the position, the linkage group position, the marker number, the marker name, which is in this case the interval between 2 SNP markers, the score statistics, and the p-values, right? So there is an issue with very low p-values in R to be stored. So if it's 10 to minus 16, it's for sure significant, right? We don't have any question about it, but we cannot be sure about the number here. So this is the limit that R goes. But we are looking at the score statistics when we define the peak, all right? So we have 355 here and 425 here. So we know that this one is major than this one, even though I'm comparing two phenotypes, but it's just to show that this same p-value here doesn't mean that we have the same score statistics, all right? Once we have this model, we can actually plot the profile and see how it looks like. So again, you have the triangles showing where the peak is, the QTL peak is. And we have these 2 on for the first phenotype, 3 for the second phenotype, and 2 for the third phenotype, right? So in this one, it seems that we have another one under this analysis at least. And of course, we can plot them together. If I say, and this is only going to be meaningful if I have more than one phenotype, right? Because if you are running only one phenotype, you're just going to see one profile, right? So this is the way we plot them together. And there is also a function for showing the supporting intervals. So this is how it looks like. You can actually include the support intervals here as well. So that's the shaded area for each QTL, right? Or something like this. It's just like types of visualizations that you might want to find more interesting for your analysis. So with the QTLs well defined, we can fit the final model and estimate, the variance components and the heritabilities and things like that. The summary function gives us these estimates, right? So the intercept, the variance components for the genetic part for the QTL and for the error, and the heritability, the QTL heritability, right? So 70%, 78, and 46% of total phenotypic variance explained by these QTLs for each one of these traits.
10 more minutes.
All right. Okay. And once we estimate these heritabilities, we can plot. There is another plot function that shows the location of each QTL, and also in these dots, right? So the proportion, the sizes of the dots are proportional to the QTL heritability. So we can see that the bigger ones are more heritable. It's just a visualization, right? And the color means the p-value, right? So the darker, the more significant the QTL is. Then we can estimate the effects of the QTL alleles using this function. And this is quick. And then we can also plot this to have an idea of how it looks like. So it actually shows all the, it plots in a loop, right? So you're gonna see all these QTL for each phenotype, right? So here is the QTL1 for the first trait, and the QTL2 for the second trait, right? Or for the first trait still. And these are the additive effects of the QTL alleles, right? So here we have the 4 alleles for Atlantic, the 4 alleles for the other parent, V18, 29, 7, and we can see these estimates going up and down, which is the deviation from the mean in the model. So here we have a mean of, for this first trait, right, of 73. So I'm adding 8 or subtracting 7, depending on the alleles that the individual has, right? And this can also be used to, look at the potential haplotypes you want to select using marker-assisted selection. So if you want to increase the phenotypic values, you want to select the blue ones, right? If you want to decrease the phenotypic values, you want to select the red ones. Since we are dealing with the mixed random effect model, we can actually estimate the QTL-based breeding values based on this fitted multiple QTL model. That's also quick, and then we can plot just to see the distribution of these breeding values, right? And then try, to figure out how I do selection here, which individuals are in the low tail or the up tail and things like that. Now, this is just to show the fixed effect interval mapping. So we also ran the permutation before, and this was the 5% genome-wide significance level, or the 95% percentile that Chris commented. And when we run these which is really quick. We can see that 2 QTLs were detected for the first trait, 1 for the second, and 3 for the, and 2 for the third trait, right? We can print, there is a print function here showing the adjusted R square, the lot score, and things like that, where it's located. And we can also use the same plot profile function to have an idea on how these peaks or these profiles look like. So I can plot the one from the random effect again, so we can have a comparison here, right? Like this, just to make it simpler. So in the random effect multiple interval mapping, we got 2, 3, and 2, right? And for the fixed effect interval mapping, we get 2, 3, 2, 1, and 2, right? So we missed the one here that seems to be true because it's also in the other traits, but we missed the one here on chromosome on linkage group 1, right? I think that's it. Maybe that's the time I have. I'm not sure.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding in Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.